We have good attendance now. Luckily, 68.
Just what's on my mind. I was counting the participants.
Participants are pouring in at the close of time.
Sartete, how was your trip? You're on mute. You're on mute, Sartete. We can't hear you.
Sorry. I'm very sorry. Yeah, it's good. As long as we came back intact, we find it is successful.
Okay, that's nice to hear. Maybe next time you can take me also.
Sure. You are the best candidate.
Thank you.
Actually, we are expecting more than 300 to attend the webinar because we have done some pre-registration.
Okay.
If I'm not mistaken, our coordinator, Dr. Kevanaldin Puya, may report it, maybe around more than 300 registered. It's not it, Mapuya, Dr. Mapuya?
More than 600, sir.
Oh, more than 600, I'm sorry. That's it.
For tonight's meeting.
Yeah, for the whole webinar.
For the whole program, okay.
Yeah,
this is a 3-day, right, Dr. Mapuy?
Yeah, it is 3 days.
Okay.
Our coordinator, Dr. Mapu, is very busy. Because he just came back from Korean.
Oh,
he's one of the NSSPO, and they did a great job there too. So with that, today also they are doing this blood donation.
Okay.
In Korean?
No, today at the college. I don't know how many students donate blood.
Sir, he went to Korean for Ranmauzong.
Probably. What else should he do in the Korean? Nothing else other than the looking for the.
I don't think he need to do that job because he is the most searched guy.
Okay.
Among women. Most wanted.
I think
most searched guy or most searching guy.
Yeah. How many participants? 118.
Now we have closed.
What about the principal, sir? Professor Zara is joined already or not? Dr. Mapuya.
I haven't seen him.
Let me call him. Let's wait for at least 2-3 minutes. Okay, I'll just call him.
Calling Sarzal Thanpuya.
Hello, Sharji.
How are you? We can't
I'm
see
good.
you. We want to see your handsome face.
$164 participants so far. $66. Thank you, Mr. Chairman. Good evening to everyone. I'm honored to welcome you all for this international webinar on integrated approach in science and technology.
Sir. Yeah. Yeah.
Sama po eh.
Yes, sir.
Sa hi principal hi. Asa on join lo tro.
Thì nghĩa là mìnhเลย.
Kan ka ship. Ka ri mai na phone kan. Phone đó là pick up thì tất luôn nè. Try that.
Satete, shall I call him up again?
Yeah, I just called Professor Kansara. He thought that the timing is 8 p.m. So let's just wait for 5 minutes, and then we once you join it, we can start it. Please bear with us. Welcome all the participants. We are about to start, but however, our principal, sir, Professor Tanzara, he forgot the timing. Actually, he thought that it will be start on 8. So hurriedly, he's preparing the laptop to join the Zoom. Let's wait for 5 minutes, and then after that, we can start once you join it. Please bear with us. Welcome, all of you. Okay, sir, welcome. Welcome, sir.
Welcome, Mr. Principal.
Thank you. Good evening, respected friends.
Unmute yourself, sir.
I already unmute. Is it audible? Hello?
Yes, barely.
Is it audible? Am I audible? Hello? Hello?
Yes.
Yes, yes,
Yes.
you're audible, sir.
Okay.
Yeah. What about me? Am I audible?
Yes.
Yes, sir.
Okay. Right. Thank you.
I think we can start. Let us start. I think we can start. Is there any other things we still have to wait? Okay, let's start it. Welcome, all of you, on international webinar on integrated approach in science and technology for sustainable future. this is a jointly webinar organized by Research Committee Pachung University College and Mizoram Science, Technology and Innovations Council, MISTIC. We are fortunate enough that we can collaborate with this MISTIC to organize this webinar. This webinar is organized to commemorate the National Science Day. Actually, it was back in 28 February. This National Science Day is celebrated annually on 28 February to commemorate the discovery of Raman effect by Nobel laureate and physicist Srivi Rahman on 28 February in 1928. Since 1987, the event has been celebrated all over the country under the nodal agency of National Council for Science and Technology, Ministry of Science and Technology. So this year, the theme is Integrated Approach in Science and Technology for Sustainable Future, which focus on fourfold integrated approach for a sustainable future, which are extended scientific intervention encompassing engineering, medical, and other institutions, extra scientific integration, which involves identification of the needs of other ministry like Jal Shakti, Railway, and among others, and extended science-driven all-inclusive approach integrating science, integrating the startups and industry. I think I don't have anything to say about the Raman effect, what this Srivi Rahman discovered, but who is Srivi Rahman? I would like to highlight. His full name is Chandra Sekhara Venkataraman, and a physicist from Tamil Nadu. His work in the field of light scattering earned him the Nobel Prize in 1930, and that, as I told, the phenomenon was called as Raman effect. And not only this Nobel Prize, in 1954, he was honored with India's highest civilian award, the Bharat Ratna. So I would like to highlight tonight's program. This is the first night, and we named it Inaugural Come First Technical Session. My name is Laram Liana, a teacher from Pachung University College, Department of Zoology, and we are fortunate enough that our program is graced by respected Principal, Pachung University College, Professor Etsal Tanzara, and the Chief Scientific Officer and Member Secretary, Mystic, Engineer Etsal Somliana, and Professor Encentil Kumar, Dean School of Life Science, who is going to give this plenary talk tonight. So the program will be welcome address by Professor Etsal Tanzara. After that, inaugural speech we expect from Engineer Etsal Somliana, our CSO. And after inaugural speech, we'll have a report from Dr. K. Vanal Dimpuya, our webinar coordinator. And after that, plenary talk from Professor Encentil Kumar, and which will be followed by some interaction from the students with the speaker. After all those things are finished, we'll call Dr. Hildala Rimpui, Assistant Coordinator, to say vote of thanks. So may I call Professor Etsal Tanzara to talk about the welcome address. Sir, please, the line is yours.
Thank you, Dr. Ramliana, the chairman of this session. Respected Chief Scientific Officer, Science and Directorate of Science and Technology, and Member Secretary of MISTIC.
Let me interrupt, sir. Can you just please increase your volume?
It's already full. Okay, let me
Yeah,
put
just
up my
a
microphone like this.
Okay, good, sir.
Respected Engineer Somliana, Chief Scientific Officer of Science, Directorate of Science and Technology, and also member secretary of MISTIC, Professor Encentil Kumar, Dean of School of Life Sciences, Mizoram University, a very well-known scientist and professor in the university, colleagues from MISTICS and Directorate of Science and Technology, and my colleagues, dear students, scholars, and friends. A very good evening to you all. I'm very happy that the college research committee, in collaboration with MISTIC, is organizing on observation of this National Science Day. Like as we know, the actual date was already passed, but we still celebrate and observe the National Science Day 2022. Uh, I'm very grateful for our colleagues from the MISTIC. Uh, uh, yes, Mizoram Science, Technology and Innovation Council, MISTIC, under Directorate of Science and Technology. Uh, and also, I'm very happy to have the eminent scientists and speakers from India and abroad, like tonight's speaker, resource person, Professor Senthil Kumar, who is my guru, actually. He's my guru, and he's a Dean of Life Sciences with a very good publication and eminent scientist in the field of biotechnology. And day after tomorrow, we are going to have speakers from United States of America, the Dr. Judith L. Pacho, who is a senior coordinator. And she used to be my senior in the Pacho University College in the Department of Zoology. And also other resource person like Dr. Lilian Pui Kolni, Lalani, and also Dr. Krishna Roluapuya Kiangte from BHU. I welcome all our esteemed, distinguished resource person for this international webinar, and I welcome all the participants, research scholars, and I wish this program a grand success, so that at the end of this program, from the sharing of the expertise and knowledge from the resource person and from the fruitful interaction after the speakers program, I hope it will be very fruitful to each and every participants. And thank you. And with that short welcome address, I end my speech. Thank you so much. And also, I apologize for confusing the time. Nowadays, a lot of webinar and activities, almost every night we have. I confused with tomorrow night program. I'm so sorry. Without such a very poor preparation, I appear like this. Anyway, thank you so much. Thank you.
Thank you, sir. We understand your problem. Beside your hectic schedule, you still can make it. Thank you very much, sir. May I now invite respected Chief Scientific Officer and Member Secretary MISTIC, Engineer H.L. Somlena, to deliver inaugural speech. Sir, the line is yours.
Thank you, Mr. Chairman. A very good evening to everyone. I'm honored to welcome you all for this international webinar on integrated approach in science and technology for sustainable future. Dr. Lal Ramdena, Chairman, Professor H. Lalthanzara, Principal, Pachunga University College, coordinators who work really hard for the success of this event, and all the members and students who attend this scientific webinar. I am extremely honored to have such a great opportunity to speak before you all in this honorable virtual meeting. As we all know that the National Science Day 2022 theme selected was Integrated Approach in Science and Technology for Sustainable Future. It focuses on the need to work in an integrated manner and in coordination for integrated team-based projects. There are two major keywords: integrated approach and sustainable future. As a science community, we must learn to collaborate and work for a common cause: the welfare of humans and nature, thus attaining sustainability, which will create a never-ending journey to create a better world. We must always remember that technology that have an environmental impact or human hazards is not recommended anymore. Together, we must make sure that the solution should be based on sustainability. Lastly, I congratulate the organizers of Pachunga University College for organizing this event, and I sincerely hope that the input given on this program will bear fruitful results. I wish the very best to all the participants, and I hope that this webinar will have a positive impact for a sustainable future in Mizoram. Thank you all. Thank you.
Thank you very much, sir. Thank you very much. We are lucky enough that we organized this program under your guidance. May I now call Dr. Kevanal Dinpuya, our coordinator, to give a brief report about the webinar. Please, sir, the line is yours.
Okay, thank you, sir. Respected Chairman, respected Principal, Professor H. Laltanzara, and our special guest, Engineer H. Lalsom Liana, Chief Scientific Officer and Member Secretary Mystic, and our resource person for tonight, Professor Encentil, my esteemed colleagues, and all the students who are here tonight, and those of you watching from our college YouTube channel. I, on behalf of the organizing committee, welcomes you once again in this international webinar on integrated approach in science and technology for sustainable future. As our chairman has already pointed out, this program is organized by the research community of Pachunga University College in collaboration with Mizoram Science, Technology, and Innovation Council. And we have invited 4 esteemed researchers and academicians from different science background, namely Professor Encentil Kumar from Mizoram University, Dr. Judith L. Pachuo from Texas, United States of America, Dr. Lalian Pui Kolni from Wildlife Institute of India, Dehradun, and Dr. Krista Roula Puyakyengte from Banaras Hindu University. And as decided by the organizing community, we set up a registration link where participants can register for the program. So far, 677 students, along with 35 faculty members, have already registered for this webinar. And as our Zoom meeting platform can accommodate only 300 participants, we opened this link only for the faculty members and the 6th semester BSc students and MSc students of Pachunga University College. And the rest of the science students joined this program through our college YouTube channel. And we make it compulsory for all the 6th semester BSc students and MSc students of PUC to attend this webinar. In each session, attendance will be collected and it will be counted as CCA attendance. So a link will be provided during or after the presentation. There you can submit your attendance. So I request each and every one of you to be there in each session. And lastly, but not the least, if you look at the history of the world, the effect of development in science and technology can be seen. It transformed the world, and it is transforming the world even now to be a better place to live in it. And it is my firm belief that a sustainable development in science and technology is our only hope for the future and for our own survival. So thank you to each and every one of you once again. And that's all from my side. Thank you, sir.
Thank you very much, Dr. Mapuya, our coordinator. We may now call Professor Encentil Kumar. Before we gave the line, it's very difficult to introduce Professor Encentil Kumar because he is the, what to say, like in very easiest way, he's the bulldozer of Mizoram University in Science Department. He joined the university in 2007, January, and now holding the position of professor in biotechnology. He got a doctorate from Bharatiya University, Coimbatore, and after that, postdoctoral fellow from Sun Yat-sen University, China. His research interests are human disease genomics, molecular phylogenetics, molecular markers, and bioinformatics, chemical, and even molecular ecology. Professor Encentil has published research papers around 150, more than 150, 3 books, 35 invited talks, so and so. He had a lot of what to say, marks, tag marks against his name. Even his impact, cumulative impact factor reached 280.7. That is very high. Recently, he got a paper in one Elsevier journal, The Lancet Diabetes and Endocrinology. That journal has an impact factor of 32.069. It is very, what to say, like great to hear that. That's a great achievement. Even we struggle to publish in one journal with one impact factor. He is undertaking a lot of research project, so it is very difficult to say. If you're giving all those introduction, it will take a lot of time. So I would like to say that Professor Encentil Kumar is a man of letters. May I now invite Professor Encentil Kumar to deliver a plenary talk. Sir, the line is yours. You can spend as long as you want, sir.
Thank you very much, Dr. Lalram Liana, for a very nice introduction. So, I'm very thankful for the principal of PUC College, Dr. Lal Tanzara, who is my very good friend. And I'm very happy to meet the director of MISTIC, Dr. Lalsom Liana, today. And of course, many of the faculty friends from PUC and the scientist friends from MISTIC, who have been very good to me over the last many years. So it is a great privilege for me to be present here today among all of you and to deliver a talk in the National Science Day celebration 2022, combinedly conducted by PUC and MISTIC. So like I mentioned, as we all know, the National Science Day is one of the very important events in the country, celebrated in all the schools, colleges, universities, and research institutions. And this is definitely going to build a lot of innovation and creativity in the young minds, especially the undergraduate and postgraduate students. And I once again thank all the organizers and the principal and the director MISTIC for this great opportunity. So I'll start sharing my screen and I'll go into my PowerPoint presentation. Thank you all. So I hope the slides are visible.
Yes, visible, sir. Sir,
Yes,
please
it
make it
is.
full screen. Okay.
Yes. Okay, so thank you very much. So the introduction given about this program, spoke about the theme of this year's National Science Day. So today we'll be talking about the epidemiology, the genetic variations, and the human diseases. So I'll keep my talk as simple as possible, and we'll also try to have some discussion at the end of the talk. So this is a very, very nice proverb, I would say, that every evolutionary process leaves their distinct mark and trail on their genetic material. So if anyone is asking us that how old are you, our actual biological answer or genetics or at the level of genomics, it should be 2 lakh years old. So the modern Homo sapiens who are now on this earth, right, is approximately 2 lakh years old. And during each stage of our evolutionary phase, a lot of information has been stored in the genes and transmitted to the next generations. And this is how the IQ level of the younger generations are always much higher than the previous generations. So the knowledge, the information, the experiences are stored in the genes and gets transmitted. So yeah, excuse me for a minute. Yes. So we'll start with the epidemiology. So what is epidemiology is basically like the studies that follow certain populations over time to observe whether a specific agent or exposure is likely to cause any disease for that matter. And here, today's talk, I'm going to touch upon cancer a little more than the other diseases. So over the years, to have an assessment at the population level, the lifestyle, the food habits, the environmental risk factors that are prevalent in a particular ecosystem that is threatening a particular population. So basically, it is also the determinants, cause or source of the disease. And the epidemiologists also study about the distribution of the diseases, the prevalency, the occurrences, the more dominant diseases or the less dominant diseases, and seasonal variations, etc. They also take into account the mortality for calculating the risk factors in terms of mortality and survival, and also it is highly population specific. So the term molecular epidemiology was coined in the year 1973 by Gilborn. So this molecular epidemiology field of research has dissolved the boundaries between traditional epidemiology and human genetics, or molecular genetics, or molecular biology. And basically this requires decisions about collecting, processing, and storing biological specimens. So the revolution in molecular biology has impacted the society, the science, medicine, and epidemiology in terms of public health, which is now a newer field emerging in the 21st century, and molecular epidemiology is on the forefront. So for any population to be studied, the epidemiological assessment has to be done first in terms of any disease, or whether it is an epidemic or an endemic or a pandemic, or even to understand the prevalence. So these are all certain common terminologies that are used in the epidemiological studies. So pandemic, we are now experiencing it as well, the COVID-19 pandemic, which is widespread globally. Endemic is constant pressure of a disease or usual prevalence in a population. So we could always say in terms of non-communicable diseases like cancers and diabetes, etc. Epidemics is an outbreak or a sudden occurrence of a disease in a population in a period of time, within a certain limit of time. So the methods that are generally used for biological variations in molecular epidemiology is, there are two types of broad studies, like case control, where cases are basically the patients and controls are the healthy individuals in the same population or in the same race, or a cohort-based study. So if cohort-based studies are focused upon a single hospital or a single healthcare center, so the patients visiting that particular hospital in a particular period of time, we term it as a cohort study. So there are various methods, biochemical or biological methods that can be used, like, for example, starting from DNA extraction to RNA to protein, or even the metabolic components. So the field of genomics, right, where the totality of all the genomic material present in an organism has grown up to a great extent due to the development of various molecular tools and methods. Similarly, transcriptomics, where we study the transcripts of mRNA that may or may not translate into proteins. So there are various, again, techniques available to study those. And the field of proteomics, the totality of all the protein component in a cell, right, also is equally important with respect to genomics or transcriptomics. So metabolomics is an intermediate phenomenon which all of us understand. So which is also playing a great role in understanding the health and well-being of a particular population. So all this combined together basically constitutes what we practice today in terms of bioinformatics, which has also gone into the pathway analysis, kind of a translational science, and it compares the difference associated with the diseases. So with respect to the human populations, right, which is going to be the focus of our discussion today. Out of Africa hypothesis has been widely accepted by scientists across the world, not only in human races, but also in various other organisms, including butterflies, elephants, and certain plant species, which has been documented and studied well. So with respect to the human evolutionary tree analysis based on the mitochondrial genome data, it has been found that the human races in the African continent is much, much older compared to the North or South Americas, which forms the New World, and the African world forms the Old World. So an excellent roadmap is now available for understanding the evolution and migration patterns of these human races across the world. So when we talk about human races, we are not, a single, having a single phenotypic resemblance. So human races have been very diverse, even though approximately about 99.8% of the genomic data has been exactly found to be the same in all the people across the world. So we differ each other only by 0.2%, but among the 6 billion bases, that is 3 billion base pairs, even 0.2% accounts for a huge variation, or the quantity of the variation is much higher. We all know that the environment influences the genotype to form a specific phenotype, and that is where human races, or the Homo sapiens, basically we have been evolving for the last 2 lakh years. And interestingly, only about 2%, or maximum about 2 to less than 3% of the genome codes for the proteins that are essentially required for our day-to-day activities. So the remaining about 97% was thought to be junk DNA until a decade ago, but now scientists strongly understand that this 97% of the DNA actually regulates the function of the 2 to 3% of the genome, that is genes that are essential for our day-to-day activities. So what is the importance of studying these variations, the genomic structural variations, the alterations in the gene at the structural level among the human races? It is because of various reasons that we understand today. It is a common phenomenon that occurs in the normal human genomes, which has been accounted for the major phenotypic variations. The environment, right? The kind of commensalism or parasitalism or mutualism that we experience in terms of climatic conditions and things like that determines the phenotypic variations. And again, this type of genomic variations at the structural level are common in many diseases. If we talk about cancers, like gastric cancer, we can easily list out about 75 to 80 genes that are predominantly involved in the process of gastric carcinogenesis. If we talk about breast cancer, we can list out easily about 50 genes that are playing a major role in different populations at different level of frequency to lead to the process of breast carcinogenesis. Similarly, diabetes and various diseases are now being implicated to the mutations or alterations that are now being found in the human genomes differently in different races. So also, these structural variations are now showing up in rare diseases. Like many families are having this type of rare diseases which are still unknown. The rare diseases runs in the families without even being known by the people in the family. And today we can even say that not only non-communicable diseases or rare diseases, but also communicable diseases, like for example tuberculosis, TB, can also, people can be genetically predisposed even to mycobacterial infection that can lead to exhibition of this TB symptoms. So in a household where 5-6 people can live in a single household, only one person exhibits the symptom of TB, whereas the remaining family members do not exhibit, even though they may harbor mycobacterium in their body. So it is because of the mutations in the toll-like receptor genes, in the immune genes, interleukin genes, etc. So this is one classical example of how Tibetans can thrive at high altitudes, because at high altitudes there is low oxygen level. So many people face difficulty in breathing or even to survive there for many years. But how are the Tibetan populations being able to adapt to those low oxygen levels? It is because of a gain of function mutation in the hypoxic gene EGLN1. Which makes the metabolic activities, especially the respiratory activities, differently in this Tibetan populations. So one very basic definition for the sake of students. There are two terminologies that people always confuse when we talk about mutation or single nucleotide polymorphisms. One is the word polymorphism itself, and the other word is mutation. So a single base change, like maybe A to T or G to C or G to A, occurring in a population at a frequency of more than 1% is termed as common polymorphism. Like it is a single nucleotide polymorphism. It is not dangerous. It is unique to the population. Over the years, the population, according to its environmental conditions, have survived. If the single base change occurs in less than 1% in a particular population, it is considered to be a mutation because it is unique, it is rare, and it may be dangerous or it may be adaptive as well. So in the Sanger technology, the sequencing technique, which is also called as the chain termination method, we all know that Frederick Sanger, who was also awarded a Nobel Prize, it can handle a single gene or only partial gene sequence. So the maximum good reach that we could get out of the Sanger data is about 900 to 1,000 base pairs, not more than that. So if we have to sequence a gene which is of about, let's say, 20,000 base pairs, we need to do 20 PCR reactions targeting the regions and 20 Sanger sequence cycles for generating the data. The alternative is, which is somewhat called as the page-by-page strategy. Whereas the alternative is all at a once sequencing strategy. So today we have next generation sequencing, wherein the human whole genomes can be sequenced within 48 hours. The bacterial genomes, whole bacterial genomes can be sequenced within a day, and the whole exomes, right, within a matter of, again, a day. So such amount of data are being generated at the sequence level throughout the world on a large pace to understand this. So as we discussed just now, so what can be sequenced? We can sequence the whole human genome, wherein we can study the structural variants, point mutations, or single nucleotide polymorphisms, copy number variations, etc. We can sequence the whole exomes. About humans, we have about 25,000, less than 25,000 genes. So the whole exomes can be also sequenced. PCR amplification, amplicons by Sanger sequencing method. RNA sequencing can sequence the transcriptomes. And various other technologies are also in the market. So when we talk about human genetic diseases, one aspect that we are now strongly believing in is our genetic constitution plays a major part of determining our lifetime health, right? So the day we are born, the genes or the DNA variants that are present can determine all the diseases and the maybe disease or well-being up to the age of 70 years, 80 years. So it is already determined and it is already coded. So that is where scientists during the years 2010, 11, they had a series of publications wherein they point out that sequencing, they have proved that sequencing of a carcinogenic gene, gene responsible for cancer, at an early stage, at a population level or at an individual level, can even predict 5 years or 10 years ahead of the person who's bound to get a cancer or other diseases. So if that is going to be a kind of an early diagnosis that could be done, the person can be alerted for the possible existence of the mutations that can lead to cancer, and the person can lead a healthy lifestyle wherein he can avoid the disease or at least postpone the disease. Instead of getting cancer at the age of 50, he or she may postpone it even up to 70 years by following a healthy lifestyle. So this type of genetic diseases, even including cancers, are most of the time multifactorial conditions. It may involve multiple genes, it may involve nuclear genes, it may involve mitochondrial genes, and these genetic disorders are responsible for an immense amount of suffering in terms of health and well-being, reduced quality of life, shortened life expectancy, and so many of the common diseases of our later life have a genetic component, their etiology. So here is one slide which tells us about why we need to understand the gene mutations responsible for various diseases, not only for early diagnosis, but also for a proper prognosis or to get a proper treatment. So this slide talks about one size does not fit all. That is very true. In the sense, percentage of the patient population for which a particular drug is ineffective. So if we come down to cancer drugs, 75% of the patients who are receiving the cancer drugs are not benefited by the cancer treatment as of today. Only 25% of the patients benefit. This is a worldwide data analysis. Similarly, metformin is one drug which is prescribed for all the diabetic patients. So nearly 43% of the patients do not benefit by taking metformin. We can move down to many of the neurodegenerative disorders. We can go to arthritis or antidepressants or asthma. So that is true for any of these diseases. So this is because the genetic component varies. The genes involved may also vary in different populations or different races for the same disease. Not only the gene differences, but also even the mutations present in the same gene may be at different locus. So obviously, this is going to change the protein component as we understand the collinearity of the gene and the polypeptide. So if the protein components are different, then the drug is not going to work with those diseased proteins to cure them or to kill those diseased proteins. So the drug is going to be ineffective. So that is where the field of biomarkers studies in medical research have gained lots and lots of significance. The developed nations have been working on this for the last 2-3 decades. So fortunately in India, in the last decade, or within a span of 10 years, a lot of research is into play, wherein we tend to study our populations, the Indian populations in general, and then we tend to understand the gene mutations and variants, and not only for early diagnosis, but for a proper treatment decision. So this, when I talk about genes involved in these diseases, it is basically a biomarker which can identify or which can differentiate between a healthy individual and a patient samples. So it may be found in the blood sample, it may be found in the body fluids or tumor tissues. It may be found in the hair follicles, along with the root. If we can remove the hair, we could isolate DNA. It may be found in other biological samples as well. So a lot of biomarker studies have been put into focus. So there is various types of biomarkers. So based on the disease state, there can be predictive biomarkers, there can be detection biomarkers, diagnosis marker, and prognostic. So prognosis is after the patient gets the disease, how does he respond to the treatment? So based on the biomolecules, it can be at the DNA level, RNA level, protein level, carbohydrate level, and of course, lipids and other biomolecules as well. And based on other criteria, like there are so many imaging markers using artificial intelligence and other things, even the X-rays or scan reports can be used as markers for diagnosis of a prognosis. There are a lot of pathological markers, especially the immunohistochemistry and other types of pathological markers used for analysis. Bioinformatics has today's students are really gifted in the sense the databases, the algorithms, the resources available for research, especially in terms of human genomes and microbial genomes, are very, very strong. The bioinformatic pipelines are very strong for analysis and interpretations. So where do these biomarkers come from basically? So as we discussed in the previous slide, it can be from gene to protein to other biomolecules. So it can be from basic laboratory test, a diagnostic lab. So many of these diagnostic labs are turning into molecular diagnostic labs. And then from electrophysiological data, from physiological data, from behavioral data, from histological, cytological, and imaging studies. So the clinical uses of this diagnostics is like it can go in a phase-wise manner. Like for example, at the preliminary stage, it can be at the risk assessment itself, even at a population level, to assess the population, whether the population, how many of these individuals are at an increased risk for cancer. The second application could be like at the diagnosis. If a person is having some problems, showing some symptoms, so it can be used as a diagnostic marker to understand that whether the person has the cancer or not. The third is once the person has got the disease, right, and then he goes to the clinic or to the hospital. So these molecular diagnostics can be used for what is the expected course, how is the cancer going to progress, what is the main cause of the cancer, so that the treatment can be given at that specific points. Then will my cancer respond to medication, for example? So predicting the treatment response. So that is basically the extended prognosis. So pharmacokinetics, to understand the state in which the drug molecule is going to act the best. And pharmacovigilance is the adverse drug reactions and how the patient is responding to the drug. So monitoring the treatment response and monitoring recurrence. So whether the disease is going to come back or not, right? Everything can be understood and can be inferred and can be tackled at the right stage. So this slide, or this information is from UMDF, in the year 2009, the United Mitochondrial Disease Foundation. They have said that every 30 minutes a child is born will develop a mitochondrial disease by the age of 10, and at least 1 in 200 individuals in the general public have a mitochondrial DNA mutation that may lead to it. So this is on a world average, right? So also, if you happen to travel to the streets of London, if you travel in a bus, you will see that every half a kilometer, there are big banners which says that every 15 cigarettes you smoke, there is a high chance of acquiring or getting a DNA mutation, a single nucleotide polymorphism in your genes that might probably lead to cancer. right? So all these types of assessments, right? Whether it is genetically inherited or whether it is through some other lifestyle or environmental risk factors, can be understood by simple sequencing information. So if we come across the spectrum of the mitochondrial diseases, right? The mitochondrial DNA mutations can lead to what type of diseases in a human system? It runs from head to toe, right? You can, so much of this information have been already implicated. About 600 disease associations in humans have been earmarked to the mutations found in the mitochondrial gene. So mitochondria is inherited from mother. It is about 550 base pairs approximately, and it it doesn't undergo kind of DNA repair mechanism, so there is no proofreading activity. So it can trace back not only the lineage, like it doesn't alone tell us the history of the people, but also it can tell about the disease and disease conditions. So similarly, the nuclear gene mutations have also been found to be different for the same disease or for various other types of cancers. Different types of variants have been reported in different populations and are unique to those populations. For example, we'll move on to the nuclear genome, BRCA1, BRCA2 genes, the two main genes implicated for breast cancer. So for the same disease across different populations, different types of mutations or polymorphisms have been reported. So this is where the drugs designed for a specific population is not going to work for other populations. So this is one study done by two scientists from IGIB, CSIR IGIB, New Delhi, Dr. Vinod and Dr. Sridhar. So they have published a paper in 2017, which says that the adverse drug reactions, right, or the pharmacogenetics of the 5-fluorouracil, the chemotherapy drug given to most of the cancer patients. So there are world reports which says that about 20% of the patients die because of this treatment of 5-fluorouracil and not because of the cancer itself. It is because they are intolerant to this chemo drug. They have adverse drug reactions. So by a simple sequencing of this DPYD gene, Sanger sequencing, just by targeting 4 exons to look at 4 variants that may determine whether a person is susceptible to the chemo drug or he is resistant, or whether he can be given. So even if he can be given, what should be the dosage? So within a matter of 24 hours, it is possible to take about 200 microliters or 0.2 ml of blood from the cancer patient, run a PCR of this DPYD gene for the 4 exons, run a Sanger sequencing, find out the variants, and we can suggest to the oncologist whether 5-fluorouracil chemo drug should be given or not. We can save the life of a patient just within 24 hours, right? After the patient has been found with cancer or the patient has been operated for cancer. So these are all the various types of adverse drug reactions that we could see, including hair fall, vomiting, and other gastric problems. So the cancer challenge, right? It is, I mean, we all know that cancer has been the second leading cause of death worldwide. 15 people die from cancer every minute worldwide, and 8 million people deaths per year. And even in the state of Mizoram also, approximately 1,700 new cases of cancer are being detected every year, and at least 700 people die out of cancer every year. So there is a huge genetic risk, not only because of smoking or alcohol or consumption of smoked food or other environmental risk factors, but the population worldwide, right, has been found that 1 in 10 adult cancers are genetic in origin, and 1 in 3 childhood cancers are genetic reasons, not because of the fault of the individuals. So this is a data from US population, because for Indian populations, we don't have a data as such till now. So for the genetic risk of cancers because of the involvement of various diseases, like, for example, including TP53 gene or other types of gene. So this much number of individuals are carrying mutations in those gene alleles, and approximately this many people living in the US are at high risk of cancer. So taking a cancer family history is very, very important to understand the pedigree analysis. So even here, based on our research finding, we found that about 43% of the gastric cancer patients are having first degree relatives with one or other type of cancers. Similarly, 46% of breast cancer, gastric cancer patients are having first degree relatives with one or other type of cancer. So almost about 50% of the cancer patients approximately are having first degree relatives within their blood with cancers, one or other type of cancer. So the level of genetic predisposition is relatively higher compared with the world population. So when should genetic testing be considered? So basically the process of genetic counseling, genetic screening using molecular diagnostic methods, and then going in for genetic counseling is very, very pertinent and important for every population across the world. So if a patient has reasonable likelihood of carrying an altered cancer susceptibility gene, then he or she should think of sequencing his or her genes to find out. So if genetic test is available, that can be so for different types of cancers, now this mechanism has been involved. So the results will influence medical management or aid in diagnosis of a hereditary cancer syndrome. So there are various genes that are responsible for hereditary gastrointestinal cancers syndromes. Like, for example, Li-Fernet syndrome is predominantly implicated for mutations in the TP53 gene. So people who are having mutations in the TP53 or inherited or acquired mutations can have a high risk of cancer of various types. Similarly, P10 gene for Cowden syndrome. So these are all the syndromes which the doctors and the clinicians can predict even without exhibiting any other symptoms, right? So these are all the different genes. These are all the DNA repair genes that have been implicated as well. So for hereditary breast and ovarian cancers, the main genes involved are BRCA1, BRCA2. So the prevalence in Western European descents are about 1 in 400 to 500 individuals, and in Ashkenazi Jewish population it is 1 in 45. So we are yet to determine for our Indian populations as a whole, and it is still in the process. So the tumor sites in families with TP53 germline mutations, you can see breast cancer, bone cancer, brain, soft tissue, gastrointestinal, or other types of cancers, or gynecological cancers. These are basically the Li-Fernet syndrome can have this. So cancers, right, not only for humans. Unfortunately, if you compare across the canines, the dogs, it has very, very high level of incidence of cancer probability than humans. Approximately 1.66 million diagnoses each year for humans, whereas 4.2 million diagnoses each year. So cancer is running across life, right? So it is the paradox and the promise of comparative oncology that we can study. So a lot of research has gone into the dog cancers as well. Another interesting feature is elephants do not get cancer, right? This was a very, very intriguing factor for scientists. Later, they found that elephants have 40 copies of TP53 gene. Humans, we have only 2 copies, right? So if these copies of genes are getting mutated, then we are prone to cancer. But elephants, because of the backup of the TP53 gene, so they are resistant to cancer. So this study has been done by National University of Singapore scientists, wherein they did a gene permitting of TP53 gene into mice models, and they found that mice models with multiple copy numbers of TP53 gene were resistant to cancer, whereas mice models with 2 copies of this TP53 gene got cancer. So the breast cancer genes associated with identifiable hereditary cancer syndromes. So for hereditary breast and ovarian cancer syndrome, BRCA1, BRCA2 genes. So if in a family, if you find 2 or 3 women with either breast cancer or ovary cancer, so they may be having mutations with BRCA1 and BRCA2 genes. So many of these genes, including the TP53 genes, are druggable genes in the sense, based on the mutations found in them, they can be treated accordingly, wherein they may recover more faster than arbitrary treatments or random treatments. So also, like there are various diagnostics available in oncology, like for hereditary cancer syndromes. These are all the possible assays that can be done in a molecular diagnostic, say like in PCR-based method or Sanger sequencing, the tools of molecular biology and biotechnology, basically. The next generation sequencing panels or exome sequencing, and so on and so forth. So for example, predictive markers or circulating tumor markers from the blood sample, or carcinomas of known primary, etc. So when we talk about population-associated diseases, there are important things to consider in terms of outcomes in relation to genetic background of the populations. So for example, at the level of disease prevention, epidemiological studies and demographic studies are very important. At the level of detection, diagnosis, and treatment, there are so many other factors to understand the quality of life also. There are many research tools and methods through public health concepts, it should be carried forward. So basically, the precision medicine, what we talk today, based on expected response of the average patient, and tomorrow it is going to be individual. It is going to be based on the individual genomic information, their environmental risk factors, their food and lifestyle habits, right? And this treatment is going to happen through those sense. So the Department of Biotechnology, New Delhi, under Ministry of Science and Technology, Government of India, have started a Genome India project, where the 10,000 whole normal healthy individuals are being sequenced now to get a roadmap of the presence of allelic frequencies for various diseases in the Indian genomes. So from the Northeast, we are involved, Mizoram University and IBSD Imphal. We have sequenced nearly about 150 whole genomes of Mizo healthy individuals. And we are also targeting few other populations from the Northeast, from Assam, from Manipur, and from other states as well. So to understand the variations and to identify variation in the genome that might be disease causing, and also to determine how individual genes play a role in health and disease. So this is the new specialty of genomic medicine, the medical discipline that involves the use of genomic information as part of the healthcare supervision by the doctors and clinicians. So when we talk about breast cancer, for example, it can be inherited. So nearly about less than 10% because of the mutations in the genes like BRCA1, BRCA2, TP53, or XRCC2, ATM, and 90% is for non-inherited or environmental factors or diet factors or random or somatic mutations. So in our study, doing a univariate logistic regression, we found the age at menarche, age at menopause, and Marshall status to be risk factors for breast cancer. The number of children or parity, breastfeeding, lactation, and for lifestyle habits, the sedentary habits or exercise was significant. Lack of exercise, basically. The sleeping hours, less than 6 hours, was found to be significant for breast cancer. Interestingly, consumption of red meat like pork and beef in excess quantity, like every day, was a major risk factor for breast cancer. Consumption of fish was also because the level of preservatives used in these fishes were found to be 10 times higher level of formaldehyde than the normal recommended doses. Basically, the fish that comes from outside the state was having high levels of the preservatives which are carcinogenic in origin. So interestingly, consumption of fish was found to be a risk factor in Mizoram for breast cancer. Less consumption of vegetables, consumption of sorghum, consumption of smoked food in higher quantities, less water intake, and the quality of oil used were all found also to be risk factors in along with consumption of sada, kaini, and tuibur. So, in relation to the family history, first and second relative breast cancer patients were found to be highly significant. Right, so patients having first or second degree relatives with cancers, any type of cancers, were having breast cancer. Right, so that value was very high, significant, and the component of inheritability. Inheritable diseases means other than cancers like diabetes or blood pressure was also significant in the population. So this tells us a general overview about the genetic predisposition of the population to various diseases. So these were some of the mutations that we found in the mitochondrial genes of breast cancer patients, and using bioinformatic tools we found that many of this affects the protein functions or possibly damaging as well. So all these are the mitochondrial genes. Similarly, with respect to D-loop regions also, which is the non-coding region of the mitochondrial genome, we found a lot of polymorphisms and mutations. So this is in terms of the nuclear genes. So CDKN2A gene was novel, that have not been reported for any other populations for breast cancer across the world. And along with it, we had a list of genes that have amino acid changes or mutations with a specific RSIDs, which are already in the clinic for breast cancer screening diagnosis, and it was also found to be significant. So these are all some of the other genes, nuclear genes, that were found to be mutated in the breast cancer analysis that we did. So similarly, our studies on gastric cancer in the Mizo population also yielded us many interesting mutations that were found in the patients and not in the healthy individuals. So we compared it with the Cancer Genome Atlas, which covers the world population. We covered the American Cancer Research Consortium data, and these are some of the variants that we found in our studies here in MZU. So this variants are found to be different from what has been reported. Even the genes involved and the variants involved were found to be different and unique. So the risk factors for gastric cancer patients, the gender, male had more gastric cancer, the BMI, extra salt consumption, excessive smoked food consumption, tuibur, alcohol drinking, and smoking were all significant for gastric cancer. Similarly, both in the univariate and in the bivariate analysis. So finally, we concluded that extra salt, through this possible mechanism of Helicobacter pylori, enhancing the Helicobacter pylori infection, leading to gastritis and hypergastrinemia, can lead to gastric cancer. Similarly, smoked food in terms of benzopyrene accumulation and affecting the cytochrome P450 pathway, inducing gene mutations on the way, and can lead to gastric cancer. Smoking in terms of this N-nitroso compounds, especially the local cigarettes, and alcohol consumptions can all lead to the possible levels of gastric cancer. So this is one study that we did with the Indian populations for COVID-19 drug response. So the drug pathway map representing the pharmacogenes functionally disrupted in the Indian population. So we had analyzed this with about 1,000 whole human genomes among covering the entire Indian states. So we found that in the drug category, the anti-cytokine or anti-inflammatory, inhibiting the viral entry, inhibiting viral replication, others. So these genes for transporters, these genes for enzymatic actions, and these target genes were all, the pharmacogenes that are potentially, I mean, these deleterious variants present in the Indian population are potentially responsible for severity of the COVID-19 disease. So this is another slide which talks about the allele frequencies of most common potential deleterious non-synonymous variants found in Indians involved in the COVID-19 drug transport. So we found many of these gene alleles that are listed here. These are the variants, basically. Those carry those. So this paper has been published in collaboration with IGIB scientists, where they were running the Indigen project, and this has been published recently in the year 2021. So this again is based on the Indian population studies. The drugs like remdesivir or leponavir or azithromycin that has been commonly used for antibiotics for COVID, right? So like chloroquine and other drugs, ivermectin, which were all used for the drugs. So these are all the genes, responsible. So these variants can influence the level of action of these genes. So just last 2-3 slides to conclude my talk. So when we do human research, especially, there are a lot of ethics involved. So the ethical principles, like it should, the researcher should have an integrity, responsibility, honesty. There should be competence with the researcher available to do the work. There should be non-maleficence. It should benefit the participants, and there should be some level of secrecy maintained. There should be an autonomy for the patients who are participating in our research. So similarly, the participants' justice should be delivered, their dignity should be protected, the privacy of the data should be there, and confidentiality should be maintained. So if any one of you are interested to know this, ICMR has already published this guidelines. So they have 12 sections where they are, the national ethical guidelines for biomedical and health research involving human participants. So this book covers all the ethics that one needs to know to work with human samples. So this is almost my second last slide. So in research, success is a cooperative endeavor. No one can succeed alone. And our success actually depends on how we work with others in a collaborative way. So this is my last thank you slide, which I attribute to Darwin. It is not the strongest of the species that survives, nor the most intelligent that survives. It is the one that is most adaptable to change. So it is very, very important that as the technologies grow, as newer tools and methods are available, we need to adapt to it, we need to employ it, we need to use it for the benefit of the mankind and the environment. So thank you all for a very patient listening, and I once again thank everyone for this, organizers for this great opportunity today I have been bestowed upon. Thank you.
Thank you, sir. That's a great presentation, sir.
Thank you, Dr.
Hope that your contribution make the world better. So it is very interesting. I do hope that even the students, though they are the graduate student, they find it useful and they find it helpful. Thank you very much, sir.
Thank you.
May I request the participants to, if you have any question, you can ask, and it is there's a request that if you have any question, you can drop in the chat. So, sir, there are some questions in the chat box. Here, one question. Let me read it out. Which gene and how many base pair nucleotide mutation causes deafness? Is molecular diagnosis facility available in Mizoram? Sir, you are me. Please unmute your.
Yes. Thank you. Yeah. So with respect to deafness, I'm not very sure as to exactly how many genes are involved, but basically these are going to be single nucleotide polymorphisms. there can be small insertions and deletions that occur, maybe up to even 20 base pair in length maximum, or mostly it should be single base pair changes. So is molecular diagnosis facility available in Mizoram? Yeah, exactly. That is our vision and mission as well. So as of now, we have been running Sanger sequencing for many diseases like cancer, various types of cancers, diabetes and TB. But very soon the private diagnostic labs are also going to come into it in the process of molecular diagnosis, wherein anyone can walk into the laboratory and sequence or screen the genes or genomes, for themselves.
Thank you very much, sir.
Yeah, thank you.
I'll just read it out another question. I think you also can read it from the chat box, sir.
Yes, yes.
And
So one size fits all. One size does not fit all method. Complex genes of cancers, will our current treatment like chemo be able to keep up with this super specific types of cancers? Yes. Like personalized genomic medicine is where now people are looking at. So based on the type of mutations that are available, not only like I mentioned, like which drug to be given at what point of time and at what dose. So even many of the clinicians, the pathologists, the oncologists, the surgeons are all now looking towards the molecular data, the genome data. So Mizoram, even though is burdened with such huge amount of various types of diseases, but one point that I would like to make on stage today is, like, the doctors, the clinicians in Mizoram are really, really very intelligent, and they are very much looking forward for research, which we rarely find it in many other states across India. So because of this, definitely I'm hopeful, we will be able to have a lot of personalized genomic medicine strategies that are maybe in the next couple of years, right? So with the collaboration and support of the clinicians and the doctors in Mizoram, especially from Civil Hospital, from Mizoram State Cancer Institute, from ZMC, and of course, doctors from other private laboratories also. We collaborate with more than 25 to 30 doctors as of today in various diseases, and we find nearly a 200% support from them. So these are some of the things that we look forward in the future. Yeah. Thank you.
thank you, sir. Thank you very much. See, another question. There is a report that consuming cruciferous vegetables can cause cancer. So is it true? What is your opinion on this?
Yes. So there was a paper published, 5 years before, exactly on the same topic, consumption of cruciferous vegetables like brassica and other, vegetables under brassica family. Is it chemo protective? Cancer protective, or is it cancer inducing? So the conclusion in that paper was, it depends on the individual's genetic variation. If their genes involved in detoxification is mutated, like GST or mixed function oxidases or cytochrome P450, then the daily consumption of these cruciferous vegetables will lead to accumulation of indoles and a lot of alkaloids and isothiocyanates, which can cause cancer. So if the detoxification mechanism, the genes are intact, and if it is working well, it is not going to cause cancer, but otherwise it can cause cancer. So scientists are now pondering whether the daily consumption of green leafy vegetables as a cuisine or as a diet in the northeastern populations, is it the main cause of increased risk of cancer? So that's one of the research question in our minds as well. So we are also looking at the detoxification genes now to understand and also estimating these levels of these secondary metabolites in these green leafy vegetables, especially to look at this phenomena.
Yeah. That's a good answer. Great answer, sir. Thank you very much. And there are another two questions around the chat box.
Yes.
Can you read that one, sir?
If cancer is caused by mutation of gene or inheritance in our hereditary or from consumption of tobacco. Sir, my question is, there is a tropic of cancer in our globe, so there is more cancer patients in this other places. Yeah. So basically, worldwide record says about 8 to 10% of the cancers are genetic in origin. And with respect to geographical locations, yes, definitely due to environmental risk factors, exposure to various pathogens, climate change, all this, including the food habits, lifestyle habits, are all major cause. And especially taking a case example of Mizo population, population being very small, about 12 to 13 lakh population, and the population inherently practicing endogamy for many generations together. At least say, Mizo population we can trace back at least to about 230 or 240 years back. So at least with this historical background information, we know that the population has been practicing endogamy. So this is another major genetic predisposing factor, in the sense, if there is a mutated allele in the population, so through a process of biomagnification, right, these alleles becomes more and more occurring in the population, or the frequency of this variants can be increased from one generation to other generation. So these are all combinedly, right, can be risk factors. Yeah, but few more years of research can only solve many of these questions. Yeah. So the next question is, what are some problems that personalized medicine can have? Okay, so that you personally foresee in the light of Sora.
Answer, sir. You
Yes.
can continue with how can we reduce cancer in Mizoram?
Yes,
that
yes.
question, yes, I think you can.
Yes. So all the questions are really very good and very beautiful. Yeah. So the thing is, I'll take up the second question first. How can we reduce cancer in Mizoram? I mentioned, in our epidemiological studies, we found drinking less quantity of water on a daily basis is a high risk factor for women for breast cancer. Similarly, consumption of fish and also large excessive consumption of smoked food, apart from the regular other risk factors like maybe exposure to tobacco smoke and exposure to alcohol and things like that. So practicing a healthy lifestyle is always one of the major factor that can help to reduce the cancer, and also having good mental state, in the sense having moving across the societal needs and looking into the requirements of our daily life in a positive approach. So in the problems that personalized medicine can have, as of now, we, in our basic research that we have done so far, we find that there isn't much, blockade for doing such type of research. That is, what do I mean is, like I mentioned, that the clinicians and doctors are very much willing for research. They spend hours and hours together sitting with us and listening to the presentations and findings that we do for them based on our genome sequence data analysis. They also give a lot of inputs that is building up our personalized genomic research in Mizoram. And at the same time, we also get good support from the patients, right, or patients' relatives. So normally, epidemiological questionnaires, it takes about half an hour to answer, and then the patient is willing to give us the blood samples, the tissue samples. So as such, I don't foresee any problems. Only thing is the resources, the manpower to do the research, right? We need more and more of young students. Manpower to basically involve, because even if 100 people work in a single cancer disease, it has that much of volume of work to be done. So there is a shortage of manpower who come into biological sciences basically, and especially take up this type of research. And another aspect is the field of genetic counseling also has to come into place now. It is a very high time. I already mentioned during my presentation that about 43 to 45% of the patients are having first degree relatives with cancers. So it is a high time for genetic counseling, right? To look into, to give a proper advice, how to avoid the risk factors, and how to plan for a better future, right? Based on the genome data or genetic data, and also advise what treatment and what type of surgery or what type of drugs to be taken at what point of time. So genetic counseling. So these are the two bottlenecks as of now, right? I hope in the days to come it can be also solved. So there is one more question. Sleeping
Sir,
lessons.
yeah.
Yes.
There are another 3 questions.
Yes.
As of now, like if possible, sir, please answer these 3 questions. Other than this, I think it's better not to take another questions.
Okay.
If
Okay.
you have, please, the participants, if you have any other questions, sir is always willing to answer you, and you can ask him personally also.
Yeah.
Just like some,
yes. Sure.
maybe he may be tired also.
No,
So,
fine.
sir,
Fine.
take this, at least these 3 questions.
Yes, sure, doctor. Sure, doctor. Yes. So sleeping less than 6 hours and mental stress will, yes. Lack of sleep, right, is one of the major stress factor because the hormone levels gets imbalanced. And so sleep is a more, very much essential in today's modernized world. And mental stress also, right? So less the stress, the more healthier and more cancer-free. So many studies, many literatures are available. So if you can just Google also, you will get many of the data that have been published across the world populations. So then coming to Genome India project, how long will it take for this project to give? Yes. So the project is already in the 3rd year now running. So it is going to end in the next 1 year. So this data is going to be publicly available for any researcher from India to take the genomes of the healthy individuals, 10,000. And the government of India is also planning to move into the 2nd phase of the project by sequencing another 10,000 healthy individuals. So this project is going to continue, and definitely this is going to help in making us a gene chip to make a coding of all the genes, the 25,000 genes in a single glass slide. So it is going to be a personalized medicine. So we will know the level of disease alleles present in the population at what frequency, and also population wise, we can take up this type of research. So how will you define healthy individuals? Okay, so it is self-declared healthy individuals. So when we are collecting the sample from healthy individuals or volunteers, so we take into account their blood pressure. We do a complete blood biochemistry, so involving about 25 blood parameters, including the blood pressure. So if everything is under normal level, including the liver function test, the protein levels, the cholesterol levels, the sugar level, and also the other HbA1c, etc. So about 25 parameters we estimate first through the project cost, and then we take up the sample as healthy individuals. But initially, when we collect the blood sample, it is self-declared healthy individuals. Yeah, doctor, I think there are no more questions as well.
Thank you very much, sir. Thank you very much.
Thank you.
It is indeed a very interesting presentation, and indeed a very interesting question-answer hours, interaction hours. Let me say that.
Thank you, doctor. It is my pleasure, and I'm very happy that the students and the participants have asked very, very beautiful questions.
Yeah, sir. If I switch on my video during your presentation, you may find me always opening my mouth just to listen to your presentation. It is a very great presentation indeed, sir.
Thank you.
So before I called upon Dr. Hilda, I would like to inform our colleague from Mystic that our PUC, we observe this National Science Day by organizing a seminar, some from our own expense and some from collaborating with Mystic. So that I just inform you that we also organize this National Science Day celebration. We observe this National Science Day by organizing a seminar. There we collaborate with like jointly organized by like a science department with the arts department. So please note that one. So before that, do our principal have anything to say? Professor Zara, do you have anything to say?
Pardon? Hello, pardon?
Yeah, you are audible, sir.
You call me?
Yeah, yeah.
Sir, thank you so much for your presentation, Sir Encentil.
Thank you, sir. Thank you, sir.
Very enlightening, very informative, and it must be very, very useful for the participants. I'm very glad, and thank you so much. Thank you, the organizers. Thank you. That's all, sir.
Okay, thank you very much. So all the participants, if you have any questions, another questions or further questions, you can always ask Professor Encentil. He is willing to answer your questions anytime. He's very helpful to the students and to the colleagues also. So I may now call upon Dr. Hilda Larinpui to say a word of thanks. Dr. Hilda, the line is yours.
Okay, thank you, sir. Respected Principal Professor H.L. Thanzara, invitees, participants, and dear colleagues, good evening to all. It is a great honor to propose a vote of thanks to all those who have helped in making this international webinar a success. Let me start by giving glory to the Almighty God for guiding us and granting us this event. I would like to thank our distinguished speaker, Professor Encentil Kumar, who honored this program with his insightful and excellent presentation. We are fortunate to have you amidst us, sir. I would like to express our deep gratitude to our principal, Professor H.L. Thanzara, for his vision, support, and constant encouragement, and for his presence in this webinar. Thank you, sir. To our chairman, Dr. Leram Liana, thank you for flawlessly gracing this program. Engineer H.L. Som Liana, the Chief Scientific Officer and Member Secretary, Mystic, the joint organizer of this webinar. Thank you for giving a prudent and a wonderful welcome speech. We are grateful for granting us the fund to organize this international webinar. Coordinator, Dr. K. Vanlal Dinpuya, thank you for enlightening us with your brief and crisp report on this webinar. I would like to take this opportunity to thank the organizers, the coordinators and the assistant coordinators for your hard work in making this webinar a great success. My deep appreciation goes out to all of you. Finally, I thank the participants and our wonderful students who have turned up in such great numbers. We do acknowledge the interest you have shown and thank you for your cooperation. Once again, I thank all for your cordial cooperation and participation. Thank you.
Thank you.
Thank you all. Yeah.
Thank you. So, sir, the students before in announcement for the students, sir, if you want, you can leave.
Okay. Thank you, Doctor. Yeah. So Dr. Zara. Yeah. Thank you.